Good morning, everybody. This is day 3 of the workshop, IBM workshop on information lens. Day 3 subject is biology and AI. Can you all hear me? Give me a thumbs up. Okay, good. And I'm Yuhai Tu. So I will be giving the introduction talk. It's going to be short and sweet, hopefully. Okay, so let's start. The first thing on the agenda is, on the subject is biology. Let's think about biology for a moment. And in particular, let's think about biology through the lens of information. By that, I mean very generally, not necessarily after Shannon, right? So definitely people have been start thinking about information before Shannon. But the example I'm going to give you is by non-biologists. So thinking about biology by non-biologists, at least not card-carrying biologists. Okay, so that's why I'm not going to tell you anything about Watson and Crick. Instead, I'll start with your hero. I think it's your hero, or the title is AKA the lesser-known work of well-known people. Okay. The first example I'm going to give you is the hero himself, Turing, Alan Turing. I don't know how many of you know that at the end of his life, he did a monumental work, in my opinion, not in computer science, but in biology. The title is called The Chemical Basis of Morphogenesis in 1952. Remember, he died in 1954, tragically. So the motivation is trying to understand basically spatial information. So how does animal have this sort of coding pattern that is, passed on from generation to generation? So what is the reason for this regular pattern? I give you an example of Drosophila fly embryo, which is not part of Turing's motivation, of course. His motivation was more about, in general, how does a homogeneous system give rise to this spatially inhomogeneous pattern, right? So he came up with the reaction diffusion equation, which gave rise to spatial inhomogeneity, starting from a homogeneous set of reaction diffusion equations. Okay, so that's example number 1. Example number 2, moving on, this is chronically, right? Is actually by another hero of computer science and a famous mathematician. He's known for his mathematics and computer science, of course, John von Neumann. You probably know that his last work, published work, is actually this book. The book was the result of a series of lectures he gave at Yale, a Silliman lecture series. So during the course of writing this book, entitled "The Computer and the Brain," he actually passed away. So the book was actually was published first, 1958, after he died. So it was sort of a very nice little book. So he wrote about the relation, the comparison between the computer and the brain. The computer, as he know by then, which have all the, I think, necessary ingredients of the modern computer, and the brain, that as far as he knew at that time, that he wrote the book, or he wrote the lectures. Moving on forward, let's go recent example. A little bit more recent example is about error correction. I think Charlie alluded to this a little bit in his talk, which I rudely interrupted, which he will give again, I think, tomorrow, hopefully. Basically, the question is, okay, if you want to replicate something, a long string of ACGT, you're inevitably going to make a mistake. Now, how much mistake is determined by the binding energy between the base pairing, right? And if thermodynamics is correct, then if everything works, then your error rate, let's say, is 10 to the minus 3, which turned out to be a intolerably inaccurate number. So you need to have some error correction mechanism to correct those errors. So here comes my third famous people for this lesser known work by John Hopfield and simultaneously by Niños. So the kinetic proofreading scheme proposed in 1974 a new mechanism for reducing error in biosynthesis, in copying, basically, right? If you have a template, you want to copy it, And you're gonna, if you make mistakes once in a while, so the idea is basically you do it again, right? So you move, you synthesize, let's say 3 base pair, then you move back, you sort of redo it again. And by doing repeating the process, you do proofreading, and then you can increase that. So that's not the end. So there's another one we're all familiar with along this line. Is actually Charlie himself. I don't know. Charlie is known for his quantum information and so on and so forth. But how many of you do know that Charlie actually did a work on dissipation error trade-off proofreading in a biology journal called Biosystems in 1979? I mean, Charlie, of course, remembers that, but I don't know how many of you know that the Charlie did work in this area as well. The reason I mentioned this is that all of these examples is that these people, they may not even mention Shannon's entropy. I mean, in Turing's case, he probably, I don't know. I mean, he probably knew, but probably not. Anyway, so this is not just necessarily Shannon's information theory, but this is about worrying about one thing in particular, all this work. What that is that how living system, biological system, actually can process information or compute. In the case of Turing, John von Neumann, is that how do the biological system compute with very sort of noisy, not reliable, and slow components, right? That's the key question, and do it accurately enough for the animal to survive. So that's the key question that asked by all these people, and there are actually really 2 questions that need to be asked. One is, how does it actually work, right? If you understand how it actually works, maybe, just maybe, you can emulate it in such a way that that in artificial systems. So that's the sort of the idea about AI today, right? For example, the brain. Another side of the coin, another question you should ask is that, okay, you can do it, but how much does it cost you to do it, right? Therefore, the thermodynamic cost of doing information processing. So that's the contribution that I think Charlie, in the context of error correction, did in the 1979 paper with respect to the kinetic proofreading scheme that proposed originally by John Hopfield. And that subject of thermodynamics of information processing will be discussed tomorrow, day 4 afternoon. Now, AI through the lens of information, right? So I have biology, I have AI. Now, unless you live in the cave for the last 5 years, I know some of you are, some of you did, you would have noticed an ongoing revolution called deep learning, right? You don't have to go very far. You go to this very famous site called the Google Scholar, and you check that, you know, this paper by Hinton and his students and postdoc about the convolutional neural network that published in 2012 in NIPS, and you look at the citation over the last 6 years. It's exponential growth. And if you look at the first few years, it's actually triple every year. The citation number per year triple. And over the past 6 years, it's more than 60-fold each year, not total, okay? The 60-fold each year by over the six years. But do we really understand deep learning 60-fold since it's first, popularized 2012? Of course not. So our understanding of the far less behind its popularity, far less. So the question is, can we really understand AI through this particular form of AI, DNN, deep learning, deep neural networks, through the lens of information? That's the question. Now, since we're talking about both AI and biology, I thought I may want to think about, we want to think about the history of the connection between biology, in particular brain, and AI, right? So let's go back in history again to look at the mother of all modern deep learning AI, right? The mother of all that is really the perceptron. So perceptron was first developed by Frank Rosenblatt, a psychologist he worked at Cornell, Navy lab at Cornell. And in 1957, he actually first sort of published a paper that proposed the algorithm and ran it on an IBM machine, 704. I don't know if anybody here knows about the 704. Probably Charlie does. He's nodding. And he actually implemented the algorithm in the hardware, so-called the Mark I perceptron in 1960. Now, in 1958, after he first published this algorithm, there was a huge, it says, it caused a heated controversial among the fledgling AI community. And based on Rosenblatt's statement, the New York Times reported an article entitled, "New Navy Device Learns by Doing," which proclaims the perception to be the embryo of an electronic computer that the Navy expects will be able to walk, talk, see, write, reproduce itself, and be conscious of existence. Embryo. He's being cautious, a little bit cautious. It's just the embryo, right? But it has all the promises. Now, of course, perception, as I said, has all the DNA of modern deep learning. Now, the question is, has this AI embryo developed to fulfill all the promises? Oh, let's look at the scoreboard, right? Versus human, machine versus human. In there, I think the machine's doing extremely well. The score is 3-0, right? So in 1997, we're from IBM, so we're very proud. 97, we beat Kasparov in chess, right? In 2011, IBM again beat Jeopardy champion, human. They beat human. And again, in 2016, by using deep learning, Google, beat the world champion in Go. It's a fantastic feat. So everybody was like, has AI finally been born? This embryo has finally developed. Oh, maybe not. Maybe not, but perhaps not so fast, right? So, okay, now we're talking about lens, going through information lens, looking at AI, looking at biology. What about, let's look at AI from the lens of the brain, right? From our own brain. So this is really a partial and growing wish list. You can say wish list or to be improved list or complaint list, whatever you want to call it. But the list is long and growing. For example, AI is just this deep learning is just a black box, very fragile, very subject to very tiny perturbation will cause a huge mistake. And our brain can learn by using very small data, one-shot learning, transfer learning, and the AI, the deep learning is really heavily supervised and data hungry. And our brain learns continuously without suffering things like catastrophic forgetting, but the current the deep learning does, and so on and so forth. You can read the list. The last one is my favorite. It's just think about pure number, right? Our brain consumes 20 watts. This much was mentioned by John von Neumann's book. In this one page in John von Neumann's book, he said 10. Well, let's give it a take. 10, 20 watt of power. But think about the computing power goes into the AlphaGo or IBM Deep Blue and so on and so forth. It's just by orders and orders of magnitude. He himself, John von Neumann, noticed that huge difference. That's the one advantage our brain he recognized. Our device is very bad. Our neurons very noisy. Our wiring is not anyway. So, but this is one advantage our brain has, which we don't really understand. Okay, now what to expect from day 3? Well, I don't know. Okay, so but I'm a naive physicist, so this is the where I come in. Before the session. So I will use Sam Cooke's song, right? Don't know about biology, don't know about history. So I come in without knowing much about biology, one subject. I come in also don't know much about artificial intelligence, right? So I think I said this to most of the audience, I suppose, is that for biology in the audience, to get to know a bit about AI, it may be useful. For AI specialists, learn something about biology, or especially the brain, it may be inspiring. And for physicists and mathematicians like me and others, we're the lucky ones, right? We got to learn both of these subjects. And who knows, we may actually, after this, help develop a healthier AI baby from this perception embryo as they promised in 1958. And as Sam Cooke continued his song, what a wonderful world this would be. So here's what I envision a wonderful world look like. This is not a wonderful world. This is a terrible old world. this old world biology, DNA, cells, and brain, they are separated from the AI. There's some connection, very vague connection, very superficial connection, but they are separated. A wonderful world has to have this thing, the information lens, because, right? So that's what we are here for. We are here to use the lens to understand biology and understand AI. I can't help noticing that I'm joining a Mickey Mouse, which is certainly a very happy thing to have. And also, that's not good enough, because hopefully by looking at the biology and AI through this info lens together, we actually create a bridge, a rainbow between the two subjects that actually will make it a wonderful world. Okay, so here's the schedule for day 3. So it's biology and AI through the lens of information lens, and I put that the subtitle that towards a wonderful harmonious world between biology, living things, and AI, artificial things. And I think all these talks are going to address either one or two or both of the subject that I talked about, biology and AI, and how they connect with each other, and how to use information lens or information as a theory, as a tool to understand either biology or AI or both, and try to find the connection. And for that, I think I pretty much, well, I am still 10 minutes away. But that's okay. As we practiced yesterday, we can take a little break before the first talk. But for that, I'm just as you. I'm looking forward to a wonderful days of talks and discussions.
Yuhai.
Yes.
Yeah. Since we got a little extra time, that pattern that you showed about the Drosophila and Turing's work, it was interesting that he approached it very much like a chemist or a biologist, and computer scientists have solved the same sort of problem under the like the rubric of the French flag problem. How do you get a bunch of processors to make 3 equal strikes?
I see.
But they looked at it from a much more computer science perspective, and he was already looking at it in a more biological way so early as 1952.
I see. I didn't know that the computer science has worked on the French flag problem, but I tell you this, I didn't say this. When I flashed this slide, what I'm saying is that it's very good to look at through information lens, and these are all pioneers. They have forward thinking, but you can say, if you're a referee, I would say this is wrong, because in development you have a gradient that guide the development. So there's not a homogeneous system to start with. So this is wrong. This is, well, partially right. There's a lot of other things involved, and there's not the only error correction. And this, well, it's not even wrong, because you don't know enough to pose the question. What I'm saying is that, yeah, in terms of biology, I mean, these things made contribution in various ways. But I think, but really most important contribution, I think fundamental contribution they have made, including yourself, is that you really get people to start thinking about how do you do computing with different type of device, right? With different kind of constraints, right? And what does it entail for in terms of designing this device, what the algorithm it can run, and what's the cost of doing that those kind of computing, right? So that's what I'm trying to come to.
Yeah. Yuhai,
yeah.
I think that one. Thank you. That was a wonderful intro to the session. You have to give
I have to stop sharing. Hold on.
No, keep it because until Talitishvi starts, we have a few more minutes. But while we ask you questions, I hope, and then we start at 11:15 Eastern. The question that I have for you is that the problem that one has when we come from the more quantitative, simplified models to more biological is that the devil is in the details, that the details in biology sometimes could be the key to, for example, in the case of Drosophila, exactly what is the network of transcription factors that get activated. And it is in the network that is the the magic of life and morphogenesis. So how do you suggest one should reconcile, the simple Ising models of biology with the enormous complexity at the detail level?
Well, I guess you have to start with the Ising model, right? I mean, Ising model was proposed. Actually, I tell you another joke that when Ising model was proposed, people was originally motivated by magnetism, right? Try to understand magnetism, right? And then a lot of theorists would be that if we were older, we would be working on it. It has very nice properties and so on. And in the field, there was actually even a name for it. I mean, of course, experimentalists and these people who know all about the details said, these guys don't know what they're talking about. And there's a name for it. It's called the Ising disease. So you have contracted the Ising disease, meaning that you're actually working on some oversimplified model. But as you and I know, that Ising model becomes not just, becomes a sort of the metaphor, or the basis for statistical physics of many interacting particles and many-body theory of statistical physics of many-body theory. That was the essence of having an Ising model. So I think in the same sense is that even though Turing may have neglected the detail of having the maternal gene have a gradient to start with, but suddenly this mechanism of a Turing pattern has been found in other contexts. And also, by the way, it's not totally wrong. I mean, in animal coding, I think people find that the Turing pattern mechanism is actually still true. So I think, what can I say? I mean, we have to start with a general question, general enough so that whatever we do will have some bearing eventually, if not in this problem, but maybe in not this specific problem, but in other problems in life. But you have to have enough relevant ingredient put in, right? Like in the Ising model, you have to have the nearest neighboring interaction, and you have the collective behavior and so on. Yeah.
Very good. So any other question for Yuhai before we go to the next talk?
Okay, let me.
All right.